In this section, we will study antigen processing and presentation by comparing MHC class 1 and MHC class 2 pathway. So, let's study these pathways through comparison. This is MHC class 1 pathway. This cell can be platelet or white blood cell. And this side shows MHC class 2 pathway. This cell can be dendritic cell, macrophage, B cell, or monocytes. The first step of MHC class 1 pathway is that viral proteins are available in the cytoplasm alongside self-proteins. They are marked by ubiquinated proteins. But in MHC class 2 pathway, any foreign material taken in by phagocytosis or endocytosis will find itself in vesicles of the endocytic pathway, collectively known as endosomes. In MHC class 1 pathway, proteasome is a cylindrical complex of proteolytic enzymes with the property of digesting proteins into short peptides. This organelle has an essential role in regulating protein turnover in all cells. Its function has been hijacked by the immune system to provide peptides for class 1 MHC presentation. Two components of the proteasome that can alter its properties so as to produce peptides with better binding properties for MHC are encoded by the large molecular assemblies with multiproteolytic activities genes that are found within the MHC region of the chromosome. But in MHC class 2 process, endosomes including the acidic lysosomes, so that various digestive enzymes can act at the appropriate pH. Process of proteolysis occurs and peptides breakdown occurs, and peptides are produced. In the case of microbial infection, the whole microbe is taken into the phagolysosome. Macrophages and dendritic cells carry many receptors on their surface, which can bind sugars or other common constituents of pathogen surfaces and greatly increase the efficiency of uptake by receptor-mediated uptake. In MHC class 1 pathway, TAP is transporter of antigen peptide. Genes are found within the MHC region of the chromosome and encode transporter proteins that carry the proteolytic fragments of antigen generated by the proteasome from the cytosol into the lumen of the endoplasmic reticulum, where they bind to the peptide binding groove of the class 1 MHC. Some viruses, for example, human papillomavirus and Epstein-Barr virus, diminish immune recognition by encoding proteins that block TAP function or peptide binding to MHC. That will be explained later in this video. But in class 2 of MHC pathway, peptides don't enter endoplasmic reticulum through TAP. In MHC class 1 pathway, the single 3-domain alpha chain associates with beta-2 microglobulin to make a class 1 MHC molecule, whose structure is not fully stable until a peptide has been bound. The efficient folding of MHC molecules around an antigen peptide requires a set of other chaperone molecules found within the rough endoplasmic reticulum. In case of MHC class 2, the 2-chain of MHC class 2 molecule forms a peptide binding groove between the alpha-1 and beta-1 domains, the beta chain contributing most of the specificity. When first synthesized, this binding is prevented by a protein called the invariant chain, which is progressively cleaved off and replaced by newly produced peptides in the endosomes. That will be explained later in this video. In case of MHC class 1, there is no invariant chain, but in MHC class 2 pathway, there is invariant chain. Invariant chain, so called because, in contrast to the class 2 MHC molecules, it is not polymorphic. It acts as a chaperone in helping MHC molecules to fold correctly as they are synthesized and then binds to them, preventing peptides from associating with the peptide binding site. While still within the endoplasmic reticulum, it then directs the transport of the associated class II MHC molecules to specialized processing endosomes, where, finally, it is proteolytically cleaved. This allows antigen peptides to bind the MHC and allows the MHC carrying the peptides to exit the endosome and go to the cell membrane. In MHC class I pathway, the Golgi complex, responsible for conveying proteins from the rough endoplasmic reticulum to other sites, including the cell surface. There is no loading compartment in MHC class I pathway. Golgi apparatus take up MHC1 peptide complex to cell surface. But on the other side, this MHC class II move into Golgi apparatus with invariant chain. Then invariant chain breaks in fragment. Now MHC2 peptide complex is found in loading compartment. LC or class II loading compartment, specialized acidic endosomes, within which peptides are loaded onto the peptide binding cleft of MHC class II molecules. The binding of peptides to MHC within this compartment is facilitated by two other class II-like molecules, HLA-DM and HLA-DO, which ensure that only those peptides with the best MHC fit are presented at the cell surface. In MHC class I pathway, TCR, or the T cell receptor, because of selection in the thymus, only a T cell, whose receptor recognizes both the MHC molecule and the peptide bound in it, will respond. This is a highly specific interaction, ensuring that cells displaying only self-peptides are not killed. CD8 co-receptor is the molecule expressed on cytotoxic T cells, recognizes the class I MHC molecule, a further requirement before killing of the virus-infected cell by the cytotoxic T cell can take place. In case of cross-presentation, some antigens break the rules and enter the class I processing pathway from the outside of the cell. Dendritic cells appear to be particularly efficient at cross-presentation, which may be of importance in trying to stimulate an immunological response against tumors. But in MHC class II pathway, helper T cell has T cell receptor, and CD4 co-receptor is a molecule expressed on helper T cells interacts with MHC class II molecules, ensuring that the T cell response, that is cytokine secretion, B lymphocytes produced response against antigen through production of antibodies. Thus, the type of T cell response that occurs is determined by a sequence of factors. Firstly, by the type of T cell, means CD8 cytotoxic or CD4 helper. Secondly, by the class of MHC, means MHC class I or MHC class II. Thirdly, by the source of the peptide bound by the MHC, means whether cytoplasmic or endocytosed. And fourthly, by the type of infection, which means viral or microbial infection. This was all about the antigen processing and presentation. In our next section, we will talk about the binding motif, immunoproteasome, immune escape, central and peripheral tolerance.